नंदिनी, विद इन 1 मिनट में इंट्रोडक्शन स्टार्ट करना है, ठीक है? 6:33 पे।
ओके सर, 6:33।
हाँ, 6:33। एक लास्ट रिमाइंडर और डाल दो आप ग्रुप में।
ओके सर।
75% अटेंडेंस जिसकी नहीं होगी उसको सर्टिफिकेट नहीं दिया जाएगा। आप एक सिंपल फाइनल रिमाइंडर डाल दो, ये 75% का मैं डालू। विद इन 30 सेकेंड स्टार्ट कर रहे हैं सर। ठीक है।
ऑफिस में ही हूँ मैं अभी, सो आई कैन इवन स्टे लेट आल्सो, नो प्रॉब्लम।
जी जी सर, जी सर। नंदिनी, अब आप स्टार्ट करिए।
ओके सर, ऑडियो वीडियो क्लियर?
हाँ, यस, क्लियर।
Good evening, everyone. This side Nandini Sharma from AgriMeet Foundation Bharat Organizing Committee, and I welcome you all to the Faculty Development Training Annadata 2025, organized by AgriMeet Foundation Bharat in collaboration with UGC and other ICR universities. It is my great honor and privilege to warmly welcome our distinguished speaker today, Dr. Raman Meenakshi Sundaram, Director of ICR Indian Institute of Rice Research, Hyderabad. Dr. Sundaram is an internationally recognized scientist whose illustrious academic journey began with his BSc Agri from TNAU Coimbatore, followed by his MSc Agri in Molecular Biology and Biotechnology from GB Pant University of Agriculture. He later earned his PhD in plant sciences from the University of Hyderabad and went on to pursue postdoctoral studies in Iowa State University and Cornell University, USA, as an Indo-US Fellow. Since joining ICR-IIRR in 1998, Dr. Sundaram has been at the forefront of cutting-edge rice research, focusing on trait characterization, molecular studies, biotic stress resistance, hybrid rice application, and transgenic breeding. His leadership and vision have shaped the institute into a global hub for rice biotechnology and molecular breeding. One of his team's most remarkable contribution is the development of improved Samba Mahusuri, a high-yielding, bacterial blight-resistant fine-grained rice variety with a low glycemic index. Today, it is one of the most successful marker-assisted breeding products in India, cultivated on over 7 lakh hectares of land, directly improving farmer livelihood and consumer health. Dr. Sundaram has also undergone specialized international training at prestigious institutions including the International Rice Research Institute, Philippines, Cornell University at Iowa State University, further strengthening his expertise with more than 200 research publications in reputed international and national journals, 50 plus books and book chapters, and numerous technical bulletins and popular articles. His academic contributions are both prolific and impactful. For his outstanding work, Dr. Sundaram has been honored with several prestigious recognitions, including the Lal Bahadur Shastri Outstanding Young Scientist Award, Hari Om Ashram Trust Award, CSIR Innovation Award for Rural Development, PN Behel Award for Excellence in Crop Sciences, DBT Award for Biotechnology Product and Process Development, and Rashtriya Krishi Vigyan Puraskar. He has also delivered endowment lectures at leading universities and continues to inspire scholars and scientists across the globe. He is a fellow of the National Academy of Agriculture Sciences, the National Academy of Sciences India, the Indian Society of Genetics and Plant Breeding, and several other prestigious academies. He has also served in editorial leadership roles, including editor in chief in the Journal of Rice Research and academic editor of reputed international journals. Sir, your visionary leadership, pioneering innovations, and commitment to bridging lab to land have not only advanced rice science but also directly contributed to the prosperity of millions of farmers and the nation's food security. It is indeed a privilege for us to host you today on behalf of all the participants and AgriMeet Foundation Bharat. I extend a warm welcome to you, Dr. Sundaram. We are eager to hear your words of wisdom, which I'm sure will enlighten, inspire, and guide us in our own professional journeys. A very warm welcome once again, sir. Over to you. The session is yours. Thank you so much.
Thank you very much, Dr. Nandini. Before I start my presentation, I would just like to know, about the listeners in terms of what is their background, so that I can fine tune whatever I'm going to my lecture. I can fine tune it accordingly.
Faculty and scientist of SAUs, CAUs, and ICR system.
Yeah, they are from which discipline, sir? What are the disciplines in which they are?
Multidisciplinary, agriculture and allied sciences.
Okay, that's fine. That makes my job easier. So I'll start sharing my presentation. Okay, just a moment.
Yes, please.
Are you able to see my screen? Of course.
Yes, yes, sir. Yes, sir.
Yeah. I need to, sorry, just share it. Okay. Yeah.
आप प्लीज इसको पीपीटी मोड पे कर लीजिए तो अच्छा लगेगा. Yes, thank you. Thank you.
Now you're able to see the screen, no?
Yes, 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 sir. Yes, sir. Able to see.
So a warm welcome to all the participants, and my sincere thanks to Dr. Ankur Sharma and also AgriMeet Foundation for giving this opportunity. What I'm going to speak today is the topic designing climate resilient rice through genomics assisted breeding. Some of you may be familiar with the term genomics. Most of you will be familiar with the term breeding. But here is a very welcome marriage of genomics and then plant breeding, which is helping us to develop climate resilient rice varieties. So my talk, sorry, my screen is not moving. Okay, my talk is divided into 5 parts. First, I'll be talking briefly about the status of rice or status of a scenario of rice in India, what kind of research that is going on, and threats to rice production, and also possible solutions in terms of how we can tackle the threats to rice production. I'll also be giving an introduction to genomics assisted breeding, a process which helping or hastening or improving breeding. And I'll also be briefly talking about genomics assisted molecular mapping, which involves development of biparental cross mapping, marker trait linkages, genome-wide diversity, natural allelic variant, genomic selection, etc. And I'll be talking about GAB applications. GAB stands for genomics assisted breeding. GAB applications for climate resilience, which includes biotic stress resistance, abiotic stress tolerance, multiple stress resistance, and also yield improvement. And I'll also talk about some new tools available to accelerate genomics assisted breeding, like haplotype-based breeding, speed breeding, gene editing, artificial intelligence, etc. And I'll also be concluding based on what I have learned in the last 28 years. And this is not something wherein I'll be teaching like a classical textbook format. What I'll be talking is what I have learned in the last 28 years after I joined the Indian Institute of Rice Research way back in 1997-98. So there is, if you have any questions, at the end of each and every part of my presentation, I'll stop there. And if anyone of you have any questions, either you can type it in the chat box or you can ask me so that I'll try to answer each and every question, whatever you may be having. So shall we move ahead with the presentation? Okay. Now, as you all know, rice in Indian perspective is life because it's a source for livelihood for millions of farmers. And at least 43% of us depend on rice for their calorific requirements. And it occupies around 45 million hectares of the total area. That is about 22% of the total area. And what is interesting for us to note is that our country has produced more than what is necessary or more than enough rice in the last, say, 4 or 5 years. This year, that is 24-25, it is anticipated a record production of 149 million tons of milled rice. And when I say rice, it's not about paddy, it's milled rice. And we are also serving as Vishwa Rakshak or Vishwa Margdarshak in terms of exporting significant quantity of rice. About 20 million tons of rice we exported last year, valuing about rupees 8,000 crores or at least $1 billion US. And this includes about basmati rice of 5 million tons and non-basmati rice of 15 million tons. And what we need to realize is that this non-basmati rice is serving as a food and nutritional security for more than 19 African countries and also Latin American countries and a few in the Central European countries. So through rice producing good quantity of rice and delivering nutritious rice, our country is really serving as a Vishwa Data or as a Vishwa Margdarshak, and we should all be proud about this fact. But if we look into the current production and future production target of rice in India, presently, as I told you, we are producing somewhere around 149 million tons with an average productivity level of around 3.2 tons per hectare or 3,200 kg per hectare. And what is a little bit upsetting is that for the last, say, around 20 years, we are not raised up the yield ceiling in rice. It is only raised up very marginally. We don't have any significant increase in the productivity level. And what is even more concerning is that right now the population is somewhere around 1.4. By 2030, the rice areas will decrease from 45 or 46 million hectares to 42 million hectares. And by 2050, our rice areas may reduce down to 35 or 40 million hectares, and population will increase even further. 1.62 billion will be the population and number of people in the country. And we need to produce almost 168 million metric tons of rice. And this rice has to be produced. There are a lot of challenges in meeting this production levels or productivity levels. Of course, population is increasing and rice yields are plateauing, which you all saw in the previous slide. And we have a rapidly changing climate, which is a real threat. I'll explain to you later. And with the changing climate, the pests and diseases are also becoming very dynamic. And we also have diverse rice growing ecologies and problematic soils. That also I'll come back after a little later. And we have to not only to deliver enough quantity of rice, but rice which is good in nutrients also in terms of more proteins, more zinc, and then more iron and more vitamins and all. And all these are to be achieved with shrinking land resources in terms of reducing rice area. And if we continue with our urbanization, what is happening presently, we may end up with only around 35 million hectares of rice by the year 2060. And water resources are also dwindling. We are already seeing that many rice growing areas in the states of Punjab are facing severe problem due to degradation of soil and water resources. And if we specifically look into the impact of climate change on rice, this is a map which has been developed by United Nations organization called Food and Agriculture Organization. What is upsetting to note is that across many intensive rice growing areas, if you look into this particular map, can you see the pointer? Can anyone confirm? Are you able to see the pointer? Okay, so many areas in our country are in the red, which means they are highly vulnerable to rising temperatures and rising carbon dioxide levels. and they are highly unstable. And most of the areas in northern part of the country will be moderately or unstable, and only a few areas are secure in terms of very lesser impact of climate change on crop yield stability. And when we consider rice as a crop, we need to understand that rice as a crop is indeed a heavy sink for carbon dioxide. That's a positive point. But the negative point is rice, as we grow traditionally under flooded conditions, wherein we impound water throughout the growth cycle to prevent weed problem, is also a major causative agent for emission of greenhouse gases like methane, which essentially is emitted under flooded rice condition. When you grow rice under upland condition, the main greenhouse gas is nitric oxide. And because of these emissions, not only from rice, but also from automobiles and also from other sectors across the globe, what we are apprehending or anticipating is that global mean annual temperature will be increasing at least by 1.5 degrees centigrade by 2050 and by 3 degrees centigrade by 2100. And what we are seeing is in the last, say, 5 years itself, temperatures have increased by about 0.5%. And this increasing temperature brings in a lot of other problems, like directly it can reduce rice yields by 1 degree centigrade increase in rice can reduce rice yield by about 10%. And it also has additional impacts like increased drought, salinization, increased incidence of pests and diseases. And as mentioned earlier, if you can look into this particular map, this shows the vulnerability decrease in production with increasing temperatures. This is the scenario what we are anticipating by the year 2100. Most areas of India are either in red or orange, being highly vulnerable, except for a few parts of China, wherein some marginal increase in productivity is expected. Mostly across India, productivity decreases can be as high as 25 to 50%. And this is a dangerous situation, and then we need an integrated approach to tackle it. Another important problem is that you'll be wondering, there are many places in Punjab, Haryana, and also in Western UP, which are producing not less than 5 to 7 tons per hectare. Why our rice productivity is still around 3.2 or 3.3 tons per hectare? This is principally because only in 50% or rather 56% of area is grown under assured irrigated conditions, wherein water is available and the crop can grow and then give its full potential. Most of the rice is being grown either in rain-fed lowland, deep water, uplands or hill rice ecosystem. And in Kerala, rice is being grown in below sea level also. And rice is the only crop which can grow little bit well, even in saline soils or alkaline soils, even up to elevation, up to about 2,500 meters, which is generally the elevation seen in upper Himalayan regions of the states of Uttarakhand, Kashmir, and also Himachal Pradesh. So rice grows under diverse ecosystem. Only the irrigated ecosystem is highly productive. All other ecosystems are not that productive. So because of this reason, our country's average rice productivity is very, very low. And what is upsetting is that this changes in climate, increasing temperatures, this is also leading to more incidence of pests and diseases. For example, in the Green Revolution era in 1960, we had only 2 major diseases, that is brown spot and then blast disease. Then the scenario entirely changed. By 2005, we had about 6 major diseases, and by 2020, we now have 9 major diseases. And the same is the scenario with insect pest incidence also. By 1965, we had 3 major insect pests, which includes yellow stem borer, gall midge, and then green leaf hopper. By 2020, we now have at least 19 insect pests resulting in yield losses, significant yield losses in rice. And part of the reason why the number of pests attacking rice are increasing is because of rapidly changing climate, increased carbon dioxide emission, and also increasing temperatures. So what is the option for managing all these things? The scope is limited in terms of only deploying conventional breeding. Most of the breeders who are present in this forum understand that improvement of multiple traits through conventional breeding or traditional breeding is not possible, and it will take a lot of time. Marker-assisted breeding has got very good scope. I'll tell you about some practical examples wherein through marker-assisted breeding, we improved the disease resistance and also yield of a few varieties. Of course, a new tool called genetic engineering and gene editing has come up. For genetic engineering, there are some regulatory issues. For gene editing, scope is very good, but IPR issues need to be sorted out, and our country has made significant headway in this regard. I'll be presenting a few slides about our own efforts related to gene editing. And finally, there is a science called genomics, which can enhance the efficiency of both marker-assisted breeding or marker-assisted selection, and also genetic engineering. We'll talk about it after some time. So I'll stop here at this point of time. Does anybody have any questions or comments for the first part of my presentation? If it is there, you can ask the questions here. If not, let me move to the next section called Introduction to Genomics.
Sir, after the lecture, there will be 15 minutes doubt clearing session.
Okay. That's fine, but I wanted it to be more of a dialogue or a multi-log.
Okay,
So
sir.
if they have any doubts, they can always ask. But if you feel that it'll be better if they ask at the end of the session, that's also fine. So, sir, shall we move to the next part of my presentation? Okay. So
Yes,
I'll
please.
talk now move to genomics-assisted breeding, which is a combination of genomics and then marker-assisted breeding. So the term genomics-assisted breeding, which is a happy marriage between marker-assisted selection and genomics, was originally coined by Dr. Rajiv Varshney, but the actual definition was given by Dr. Pallavi Sinha through a paper published in Theoretical and Applied Genetics. According to this particular paper, genomics-assisted breeding is the integration of genomic tools for improving the efficiency of plant breeding. This approach leverages advancements in structural and functional genomics to identify genes or alleles associated with desirable traits and accelerate the transfer to elite rice lines. So what are all the key components of genomics-assisted breeding in rice? Of course, all the things starts with molecular mapping. Even in genomics-assisted breeding, molecular mapping is an essential component. Molecular mapping means mapping major genes and also mapping QTLs or quantitative trait loci. Quantitative traits are those traits which are controlled by multiple genes, some with major effect, many with minor effect. And then comes marker-assisted selection. Once you identify a specific gene or its location on a particular chromosome with a linked marker or a gene-specific marker, then we can use that information for marker-assisted selection to develop varieties having that particular target gene. Genomic selection is something at a higher level, wherein just not single markers, but markers across the whole genome we utilize to identify the best genotypes, which we can advance after segregation. Then another concept called allele mining is coming. That also I'll describe it later. Genomic editing is also an interesting topic. I'll also describe that a little bit later. If you look into steps in genomics-assisted breeding in rice, what we generally do is we'll generally have a mapping population which will be segregating for a particular trait. That trait will be going for large-scale phenotyping, high-throughput phenotyping. Then that population will be subjected for genotyping using different kinds of markers like PCR-based markers like SSR markers or microsatellite markers. Nowadays in crops like rice, very high-throughput markers called SNP markers are also available. And this population can also be subjected for genome-wide association study. And all these efforts will result in development of very good markers for each and every trait. And using these markers, we'll be subjecting them for marker-assisted selection. Then we'll be improving the population or a particular variety for one or two traits. And then they'll be subjected for field testing and validation through multi-location trials. Then they'll be released for commercial cultivation. If anybody is interested, in this book chapter written by Dr. Sinha, all the details are there. If required, I can share a PDF copy of my presentation to Dr. Ankur Sharma. If anybody is interested, you can get a copy of my PDF copy of my slides from Dr. Ankur. Now coming to genomics-assisted breeding, which is the crux of today's talk. If you look into genomics-assisted breeding, we need genetic resources that can be core collections, donor identifications, or a set of germplasm lines. Genetic resources can also include biparental populations, diverse unrelated panels, magic populations, and NAM populations. If anybody is interested, I can explain it to them in detail through email. What are MAGIC populations or NAM populations? MAGIC is multi-parent advanced populations. Nested association mapping populations are NAM populations. Once you have genetic resources, then you go for trait discovery using genomic tools like SNP markers or through expression analysis. Through what we do is we do go for linkage mapping and then QTL mapping and also association mapping. And we develop PCR-based markers or sequence-based markers like single nucleotide polymorphism markers, which are also called as SNPs or SNPs. Then, using these tools, we go for trait improvement through marker-assisted selection, which includes marker-assisted backcross breeding, marker-assisted forward breeding, marker-assisted recurrent selection. Then we also, wherein we'll be manipulating one or few genes. Then through gene pyramiding, we'll be improving several genes or their alleles. And then we'll also go for multi-trait selection. If you want to improve the genome as a whole, we'll be going for genomic selection. If you want to improve the alleles for a particular set of genes for a particular trait, we can also go for haplotype-based breeding. And if you look into the applications of genomic-assisted breeding for rice improvement, one important thing, this is something wherein breeders will be knowing a lot. There is something called as genetic gain, which is a direct function of the selection intensity, selection accuracy, and genetic variation. And genetic gain is inversely proportional to generation interval. This is a classical genetic gains equation. According to this equation, either by increasing the selection intensity or by accuracy or by genetic variation and by reducing the generation interval, we can have better genetic gain so that population we can improve for multiple traits across different seasons. So if you look into what all the applications of genomics-assisted breeding, first of all, it increases breeding efficiency by enabling faster and more accurate selection of desirable traits. Second, it improves precision by targeted integration of specific genes or QTLs or multiple genes by minimizing the introduction of undesirable traits from the donor lines. There's a concept called linkage drag. Whenever we try to introduce one trait, multiple traits will integrate from the donor parent, and then that leads to something called as linkage drag or a donor genome effect that is not desirable. So through genomics-assisted breeding, we can precisely improve only the target trait, not the other loci will get introgressed. And finally, it can lead to enhanced genetic gain, wherein breeders can identify and combine desirable traits more effectively, leading to faster genetic improvement. And genomics-assisted breeding is the only tool which we can use to quickly develop climate-resilient rice varieties. What I'm talking about rice, it's also true for other crops, wherein genomics-assisted breeding as a tool will be helpful to manipulate or improve multiple traits or multiple genes or their alleles, and so that we can ensure food and nutritional security in an era of climate change. Now, I'll be moving to the next part, talking about genomics-assisted molecular mapping, wherein we can talk about different populations. Are there any doubts or any questions anybody would like to ask regarding the first two parts of my presentation? Okay, let's move ahead. Talking about how genomics-assisted molecular mapping has accelerated, I'll tell you some case studies. Initially, when only a set of PCR-based markers were available, they are called as SSR markers or rice microsatellite markers. That's why they are called as RM markers. We were unable to go for quick identification of candidate gene. We were able to undertake molecular mapping, but the time scale for molecular mapping to gene identification almost took about 4 years. Another important thing is the development of identification of 2 major QTLs for BPH resistant gene. This effort also took almost 4 years because the molecular mapping was not done through genomic tools like SNP markers and also with transcriptome analysis. So it took almost 4 years to identify the candidate genes or the actual genes which are responsible. But we at IARR, we identified a gene called as XA33, which confers bacterial blight resistance that was introgressed from a wild species of rice called as Oryza nivara. When we did course mapping, immediately we deployed a set of SNP markers, and we were able to identify a candidate gene called as serine 3 receptor protein kinase. This we are able to do it within a time period of 2 years. This was way back almost 10 years ago. But recently, we were able to identify a candidate gene called as VPH33 in the background of rice. And through genomics tools like transcriptomic proteomics and also SNP, single nucleotide polymorphism-based fine mapping, we were able to narrow down the candidate gene and also transfer this particular trait very quickly into elite varieties like Samba Masuri. And the important thing is that for identification of the candidate gene, that is a heat shock protein gene, it just took us 2 years. Another example is classical work of one of my PhD students called Dr. Pragya Sinha. She was trying to undertake molecular mapping of BLB resistance or bacterial blight resistance from Oryza officinalis line. Oryza officinalis is also a wild species of rice. Using SSR markers, she was able to identify that the gene is present on chromosome 11 on the long arm. But just with around 7,000 SNP markers, within a period of within 1.5 years, she was able to identify that the candidate gene is encoding an NBRC domain containing protein. And based on the candidate gene polymorphism, she was also able to develop a functional marker. And similar is the story with regard to identification of a major gene for neck blast disease resistance from a wild relative of rice called as Oryza glumipetula. In this case also, course mapping took a lot of time, wherein a major QTL was identified on chromosome 3 just by using genomic tools, which involves both the genomics, transcriptomics, and also proteomics, mainly involving SNP single nucleotide polymorphic markers. We were able to identify the candidate gene, which is a malactin serine threonine kinase gene controlling blast resistance in Oryza glumipetula. And the gene is also known PA68. And this was done within 1.5 years. And even more interesting is identification of a major QTL associated with tolerance to both brown plant hopper and then the whiteback plant hopper. And in this regard, the whole exercise just took about 1.5 years. And very recently, my colleague Dr. Prakasam and team has identified major QT all on chromosome 11 for sheet blight tolerance. And he was also able to identify the candidate genes and develop SNP markers, which can be used in breeding. These SNP markers are part of the gene, so they can be easily used for marker-studded interrogation of this particular QTL. And since these are functional markers, there will not be any recombination, there will not be any false positive. They are used for breeding. And this process took only 1 year. So possibly, if with increasing availability of single nucleotide polymorphism markers and other tools, possibly this process of molecular mapping can even be done within 6 months also, once the population is stable and then already been genotyped or phenotyped. Then my colleague Dr. Jyoti has identified through association mapping by analysis of 181 genotypes, diverse genotypes. What she did is that she sequenced all those genotypes, identified about 35,000 SNP markers polymorphic among the parents and also the genotypes. So these markers were analyzed for the co-segregation pattern with regard to culm strength or the stem strength. And she could identify at least 3 major QTLs called QSM 2.1, QLBW 2.3, and QBS 12.1. And this kind of studies offer a new scope wherein, even though you may not have a mapping population, just by analyzing diverse genotypes and analyzing the polymorphism with regard to the trait, and through SNP markers or genotyping by sequencing, you can associate genomic loci with the target trait. And like that, many other good examples are also there in rice. Now coming to an important topic called as genomic selection. Genomic selection is done in 2 steps. The first step is to make crosses between 2 parental lines, one differing for the trait, other parent is a neutral parent. Then generate mapping populations, which could be recombinant inbred lines, backcross inbred lines, or any other segregated but fixed population. Then these populations, you select a few offsprings and you go for subjecting them for genomic selection, wherein using a large number of SNP markers, at least 1,000 or more. Generally, the numbers runs into several thousands. You also, in addition to phenotyping, genotype all these mapping populations, this ones, And then after making the identifying the suitable market trait associations, then you go for fresh crosses by identifying suitable parents. And then after crossing the parents, you can predict that you'll be getting better ones. But this is something based on modeling called as genomic modeling, wherein each and every loci will be given a value called genomic estimated breeding value. Based on genomic estimated breeding value, we'll try to introgress multiple loci, not less than at least 100 or 200 into each and every line. And this process takes some time. First, we have to develop a training population, and another population is called as breeding population. So all these things will take a lot of time. And the key for genomic selection is identifying the genomic estimated breeding value for each of the locus or each of the marker locus which you have identified. And this is generally effective for polygenic traits controlled by multiple genes like yield, drought tolerance, and resistance against a few pests. These are all controlled by minor loci or multiple genes, which each having smaller effects. And the key is estimating the genomic prediction based on genomic estimated breeding values. And this helps in identification of superior parents and also segregating progeny through genomic predictions. And simulation of breeding program helps in optimizing the genetic gain. Of late, a new tool called as integrated genomic selection has come, wherein you don't just do training population and then breeding population. All these are combined with tools like speed breeding, which can accelerate the number of generations. You also use tools like machine learning, bioinformatics, artificial intelligence, and integrate that with high throughput genotyping so that very quickly we can do integrated genomic selection. Of late, some progress is coming from crops like maize. In crops like rice, integrated genomic selection is still under standardization. But typical example of integrated genomic selection is in animal breeding, wherein it has been noticed that in several European and then Western countries like USA, Canada, they have improved the milk yield of cows by at least about 40% by identifying the right set of parents for crossing, by developing analyzing the crossing data and identifying the pedigree data, correlating the pedigree data along with the genotype, and then identifying the right set of parents for crossing. As far as IRR is concerned, we have developed a lot of training population, which we are using it for model training. And using the training population data, we develop a training prediction model and identifying genomics estimated breeding value for multiple traits or multiple loci. And using that data, we are going for line development, wherein only selected lines are crossed and then mapping populations are developed. And we are also validating these lines, whether they really have higher yield or not. That's also going on. And in this process, we are also able to develop new set of breeding lines. And all these are presently under validation. We don't have a real success story regarding genomic selection, but this is a work in progress at IRR. And what we also identified is that with the help of a statistician for genomic prediction, the role of statisticians are very, very vital. He has identified that among the various statistical prediction models, he has estimated that artificial neural network model involving genetic algorithm is the best in terms of getting a better correlation between genotype and phenotype. This specifically for grain yield and a low nitrogen. This was the training data. When we validated that data with our breeding population, we could get an excellent correlation of more than 0.7%. And as a first step, we have started implementing genomic selection in our All India Coordinated Research Project on rice, wherein in 2023, we got 1,185 lines. In 2024, we got 1,202 lines. All these were subjected for fingerprinting using 1K RICA SNP chip panel. And we have the phenotype of these lines for multiple traits like yield, grain yield per 10 meters square, grain yield per line, number of grains per line and all. So this data is being used to develop a genomic prediction model. And also for each of the SNP markers, we are trying to get the GEBV value, genomic estimated breeding value, which will be used for developing better breeding materials in our All India Coordinated Research Project on rice. I stop with regard to genomics-assisted molecular mapping at this stage. But if there are any questions, you can ask. Otherwise, shall I move to the next session, which will be interesting because there will be a lot of interesting pictures showing the development of varieties by application of genomic and then molecular tools. Can we go ahead? Okay.
Yes, Dr. Saab, yes.
Yeah. So if you look into the biotic stresses which are affecting rice, there are a lot of insects like stem borer, leaf folder, brown plant topper, and gall midge. There are a few diseases like blast, bacterial blight, sheet blight, RTV false smut, and nematodes and weeds are also a major problem in rice. I put stem borer, leaf folder in red color, BPH and gall midge in green color, principally because we don't have any known sources of resistance either for stem borer or leaf folder. Most of the sources are only tolerant, but for BPH and gall midge, we have got very good resistance sources, and then at least 40 genes have been identified with regard to BPH resistance. And for gall midge also, we have identified more than 11 genes. Same is the story with regard to blast and bacterial blight, which are depicted using green color. We have got many number of genes which we can use it in breeding. But for traits like false smut and all, we don't have good resistant donors or host plant resistance. So resistant breeding may not be possible at this stage with regard to these stresses. But as far as resistance breeding is concerned, most of the first efforts during Green Revolution or immediately after Green Revolution try to transfer only single genes, conferred resistance. They are effective till some point of time, but at a later stage, we found that they are breaking down. So, the preferred choice is that according to a changing climate, we need to deploy the right set of genes in combinations through gene pyramiding. And genomic tools help us to go for fast tracking gene pyramiding. This we'll see using some examples. Some of you might have heard about a variety called Sona Masuri or Karnool Masuri. The original name of that variety is called BPT 5204. And it's also popularly called as Samba Masuri because Samba is a season. It's called as late Kharif season, which is very popular season for rice cultivation in South India. Samba Masuri has got higher yield, at least about 5 tons per hectare. It gets a premium market price because of its medium slender, short slender grains, and it gets a higher market price across South India. For example, in the state of Telangana, even the government supporting price is 500 rupees higher per quintal for Samba Masuri or any other medium slender grain type variety. So they are very good in head rice recovery and also very good in marketing price also. But one of the major problems of Samba Masuri is that it is susceptible to most of the known pests and diseases and also to all the abiotic stresses which are known so far. So when I joined IARR, a group of farmers, particularly from Godavari districts and also from Karnool districts of Andhra Pradesh, they came and represented that they are Samba Masuri farmers. They used to get very good profit by growing Samba Masuri. But for the past few years, starting with 1994, the crop is severely devastated by bacterial blight. They wanted some solution. So my team here at IARR, along with Dr. Ramesh Sonti's team, they are at Center for Cellular and Molecular Biology, we devised a marker-resisted breeding strategy wherein we, through backcross breeding, we wanted to transfer 3 major bacterial blight-resistant genes. We selected these 3 genes called as XA21, XA5, and XA13. This simply because these genes are known to be very effective in combination and can confer durable resistance. So at each generation, we subjected the plants for selection for the target genes, and then later for the background selection. If anybody is interested in background selections, this time is not there, but I'll not be able to explain it, but at a later stage, you can contact me. I'll be able to explain it. And information is also available in one of our papers, which I'll be sharing shortly. So this process was repeated for till BC4 generation, wherein plants having all the 3 genes were identified, they were selfed, and homozygous lines having all the 3 genes were identified, and they were further selfed to narrow down into at least about 5 lines, of which 1 line was subjected for All India coordinated trials. And finally, one such line was identified not only to have a high level of bacterial blight resistance, as you can see here. This also had the same fine grain type and excellent cooking quality. Dane dane alag hai, almost like basmati, like this thing, you can see it. And this also had a higher yield. And this was released as a new variety called Improved Samba Masuri. It's being grown in at least 900,000 hectares or 9 lakh hectares presently. And it also has been observed to have a lower glycemic index. Glycemic index is something which indicates how much glucose will be released rapidly into the blood after eating rice. As you all know, eating white rice or milled rice is one of the main reasons for increasing type 2 diabetes in the country. We now have several varieties, including Improved Samba Masuri. I didn't breed for low glycemic index, but by chance it happened to have lower glycemic index. So a lot of interest is there for this variety. Even in areas as far as Uttar Pradesh, this variety is being grown. And as you can see here, one advantage with Improved Samba Masuri is that it matures at least 7 to 10 days earlier than Samba Masuri. And the crop has got a very good stand. It doesn't lodge even in heavy rains or winds. And if anybody is interested how we develop Improved Samba Masuri from Samba Masuri by transfer of bacterial blight resistant genes, you can refer to this paper. It is available freely online, and complete nitty-gritty is provided in this paper. And as you can see here, under conditions of cyclonic weather, which generally happens in coastal Andhra Pradesh, you can see on the left side, we have got Improved Samba Masuri, which is totally free from bacterial blight, which is maturing normally with good number of grains. On the right side, in the same field, we have got Samba Masuri, which is totally affected by bacterial blight. And you can see only a few grains here and there. Most of the grains are chaffy because of the bacterial disease. Now we have developed a version of Samba Masuri variety with bacterial blight resistance. But we all know that it's also susceptible to many other diseases and pests like blast, brown plant hopper, and also abiotic stresses like drought, submergence, salinity, and all. And then realizing that Improved Samba Masuri is not the end target, we need to develop better versions as compared to Improved Samba Masuri, retaining the bacterial blight resistance there. So we went for an intensive crossing program involving back crossing and also limited pedigree crossing in order to add value to Samba Masuri. So through this particular effort, for the last, say, about 10 years, we have developed several better versions of Improved Samba Masuri, which already has XA 21, 13, and 5. For example, we have developed other bacterial blight resistant version by adding a gene called as XA 38, called as DRR 53, in addition to 21, 13, and 5. This variety retains a better spectrum of resistance or a more durable resistance. And we have developed a blast-resistant version of Improved Samba Masuri, which combines bacterial blight resistance and blast resistance, having 2 genes. So this was also released recently as a variety called DRR 62. Then we have got breeding lines having BPH resistance called, those lines are under evaluation. And Galmage resistant, this year one variety has been identified for varietal release. We are waiting for CVRC notification. Then very tough traits like salinity tolerance also, we could transfer genes like Saltol and PUP1, and then we have developed varieties which can grow under lesser soil or grow under salinity condition, retaining the bacterial blight resistance and then fine grain type of Improved Samba Masuri. And my colleague Dr. Neeraja and Dr. Jyoti have transferred trait of high zinc, low GA is already there, and developed a variety called DRR 48, which not only has bacterial blight resistance, but also better nutritional quality. And finally, my team has transferred some of the yield enhancing genes like GN1A, SCM2, and OSSPL14, which are available in Japonica rice donors. And then through this, we have developed a better yielding version of Samba Masuri, not only having higher yield, but also bacterial blight resistance, retaining the 3 bacterial blight resistant genes. We have also developed submergence tolerance and drought tolerance lines, which are under various stages of All India coordinated trials. This is not an effort just by a single person. We have got a team of at least 12 scientists involved in IARR. And we also have a team of several research scholars who are working with me. And this is a collaboration involving CSIR, CCMB, several ICR institutes, and it also involved funding from not only from ICR but also from CSIR and also DBT. And this particular effort is an enormous effort which has taken more than 15 years, wherein starting with just Samba Masuri, we are now at least about 5 different versions of improved Samba Masuri, like DRR than 58, DRR than 60, 48, 53, 62, and 75. And they are all doing really well in farmers' fields. For example, this is a photograph of DRR than 53. As you can see here, it has got outstanding resistance against bacterial blight. And since we didn't opt for a complete backcross breeding, we opted for a backcross based pedigree breeding. We could get 10% higher yield as compared to Samba Masuri. And further details, you can find it out in this paper. And other example is DRR than 62, wherein we are transferred along with bacterial blight resistance, which is evident here. We are transferred blast resistance also. As you can see here, ISM is totally susceptible to blast disease, while the introgression lines are showing strong blast resistance. DRR than 62 is also just like Samba Masuri, same with excellent medium slender grain type. And further details can be had in this particular publication. Then another major breakthrough is development of salinity tolerance. There is a major QTL called as Saltol, which is a large region of almost around 1.5 MB. So we transferred this whole region using genomics-assisted breeding, using strip markers. So very quickly, within a time period of around 2 years, we were able to convert improved Samba Masuri into DRR than 58 by quick transfer of Saltol gene from a donor called as FL478. These lines not only have tolerance at seedling stage, even at field stage also. This is the way Samba Masuri grows under heavy salinity and alkalinity, while DRR than 58 is growing very luxuriously. And this is one of the varieties dedicated for the nation by Honorable Prime Minister. Another one which was dedicated by our Honorable Prime Minister as climate resilient variety is DRR than 60, wherein the background of improved Samba Masuri, we transferred a major gene called as POP1, which can take up phosphorus better from the soil, so that even with 40% lesser dose of phosphorus, it means they require only 60% of P fertilizers. These varieties can grow really well. And this variety is also becoming increasingly popular across the country. And this is another flagship development, wherein in the background of improved Samba Masuri, by crossing it with CSR 27, we have developed a version which is having higher grain zinc with not less than 24 ppm, but also low J, which has come from improved Samba Masuri. And this has now been licensed to a private company in addition to distribution to farmers. This private company takes paddy from us and they mill it, and then they are marketing it as a rice called as Glycino rice, which they are recommending it. Many hospitals are recommending this Glycino rice for diabetic patients. And the holy grail in improvement is yield. Yield is very, very tough to improve, but this is how Samba Masuri looks like. We are by transferring 2 genes called as OS-SPL14 and SCM2. SPL14 increases the panicle branching. It's called as panicle branching gene, or SPL. It's also called as wealthy farmer's panicle. So it increases the grain number indirectly by increasing the panicle branching. Another gene is called as SCM2. When you got a strong grain number, that should also be supported by a strong culm. So this is a newly released variety called DRR than 75. Last season, we had had a dharna by farmers who wanted more seeds. We distributed almost 12 tons of seeds. Still farmers wanted it. And then this variety is doing really well in farmers' field. And it is completely resistant to lodging. Heavy cyclonic winds, severe rains also, this variety will stand. And it retains the premium grain quality of Samba Masuri. And also bacterial blight resistance. And the same thing, we also did it in another variety called MTU1010, which is a long slender grain type. In this also, as you can see, MTU1010, after transfer of these yield enhancing genes like GN1A, OS-SPL14 and SPL2, has got more grain number per panicle, which is evident in this picture also. And my colleagues have developed by transferring genes like Sol-Tol and also terminal stage heat stress tolerant genes. They have developed a better hybrid called DRRH5, which tolerates extreme saline conditions, and a variety called DRR52, which has got terminal stage heat stress tolerance. And another important noteworthy development is a work done by my colleague, Dr. Jyoti Badri, wherein she has not improved just 2 or 3 traits, just like the way my team has done for Samba Masuri, wherein multiple traits were improved. She has improved at least 3 or 4 different traits like blast resistance, BLB resistance, tolerance to drought, and also tolerance to brown plant hopper. And she has developed an improved version of the popular variety called Krishna Hamsa. And DRR 79 not only is highly tolerant or resistant, high yielding, and its rice grain quality is also very, very good with very good milling recovery. And a few papers highlighting this development are represented here. And this is another typical example wherein success of marker-assisted genome breeding or genomics-assisted breeding shown here. Regular Swarna under conditions of seedling stage submergence generally cannot grow beyond 40-45 days. It totally dies. But this is a case wherein a gene called as sub1 from an Indian landrace called FR13A has been transferred into Swarna. And the work was done by my colleague called Dr. Neeraja when she was at IRRI. So this is sub1 Swarna. As compared to the condition here under submergence, this has totally recovered, and then near normal yields it is able to give. And this is another variety which has got terminal stage drought tolerance by transfer of a QTL called QDTL 2.1 and 3.1. DRR 42 has been developed. As you can see here, original IR64 is totally susceptible to drought with a lot of leaf rolling. DR 42, even after soil is showing some breakage because of very low water conditions, it's still able to grow. These are all success stories of marker-assisted breeding, wherein we have achieved by our collaborations with IRRI and other partners. And other typical example is equally good work has been done in terms of applying genomic tools for breeding by Dr. A.K. Singh's group at Indian Agriculture Research Institute. Their work is mainly focusing on Basmati varieties. For example, they have developed a Basmati variety with excellent grain length and also cooking quality called Pusa Basmati 1847, 1885, and 1886, combining multiple bacterial blight and blast resistance genes. And these two are the major stresses limiting Basmati productivity. And particularly blast disease occurs, farmer has to spray a chemical called tricycloazole, which results in rejection of Basmati seedlots because of pesticide or fungicide residue. So by development of these bacterial blight and Basmati varieties through marker-assisted breeding or genomics-assisted breeding, our Basmati is now foolproof in terms of diseases and other things. And for the first time across any country in the world, Dr. A.K. Singh's group has also developed a herbicide-tolerant variety. This is from a natural mutant of Nagina 22. They have transferred this trait, and when a herbicide called imizatapir, which belongs to imizodolidine group of herbicides, original variety cannot stand it, totally killed. Wherever that gene is there called the Robin gene, which is a confers tolerance to herbicide, this can grow luxuriously. And as you can see here, this is the advantage of marker-assisted breeding. And if you look into it, through marker-assisted breeding or genomics-assisted breeding, a total of 282 rice varieties have been released, either through CVRC or SVRC. CVRC stands for Central Varietal Release Committee, and SVRC stands for State Varietal Release Committee. And this includes 31 hybrids and 198 varieties. And if you look into a total of rice production, partly because of release of climate resilient varieties, our rice production has increased tremendously just in the last few years. For example, by 2017-18, we were able to produce only 112 million tons of milled rice, but the number has increased to about 138 million tons by 2023-24. And by 24-25, we are able to produce at least 149 tons of rice. And this is despite both normal condition and abnormal monsoonic conditions. For example, in the year 2021 and 22 and 22 and 23, the monsoon was not normal. It was below normal, but still because of release of these climate resilient varieties, either in terms of durable pest resistance or disease resistance or abiotic stress tolerance, we were able to see that we are able to get climate resilience. And what is important is that just in the last 4 years, 44 varieties have been released, and they already started occupying more than 10 million hectares, and each one having a yield advantage of 1 to 1.5 tons per hectare under either biotic or abiotic stresses. And together, these varieties have added not less than 1,000 crores of rupees. So I'll stop here in the, with regard to genomics assisted breeding applications for climate resilience. Now let's just see 1 or 2 important tools wherein we can accelerate genomics assisted breeding or marker assisted breeding. These tools include haplotype based rice breeding, wherein for each gene, we generally consider only 2 alleles, favorable allele and then non-favorable allele. Through intensive sequence analysis and also functional genomics, we have identified that there are more than 189 genes which are affecting grain yield and 63 genes. For each one of these genes, we now have several alleles identified, several favorable alleles identified, and 1 or 2 non-favorable alleles also identified. And now the task is to identify which gene has got how many favorable alleles and then which donor has got it, so that all these favorable haplotypes can be transferred to elite rice varieties. For this, my colleague Dr. Jyoti Badri has got an ambitious program funded by DBT, wherein at least about 20 yield-related traits she is trying to transfer wholesome into the background of some of our elite rice varieties. And she's halfway through this effort. Maybe in another, say, 3 or 4 years, you may hear that she has transferred at least 20-25 major genes of the favorable alleles into elite rice varieties, through which she is able to improve the yield and also other plant types like stronger culm, better pest and disease resistance and other features. And another important development is called as speed breeding. Generally, under normal conditions, rice can grow. We can grow rice only for 2 seasons in a year, because rice crop on an average takes anywhere between 130 to 170 days. So maximum of 2 seasons only we can take up in a year. But by carefully controlling the day length, for example, rice is a short day plant, and by controlling the number of period in which the light will be there, and also by controlling the temperature, humidity, and through immature seed harvesting, and also by manipulating the light intensity and quality, we can theoretically take up not less than 6 seasons in a year. And such facilities are called as speed breeding facilities. For example, for rice, we have got speed breeding facilities at IRRI South Asia Regional Center at Varanasi, and also at few other centers like Rice Research Station Navsari, and also at National Agri-Biotech Institute Mohali we have got. In my institute, we will shortly develop a speed breeding facility costing not less than 12 lakh rupees. And we have published a paper together with IRRI, wherein we have developed a very, very comprehensive speed breeding protocol for Indica and Japonica rices, through which, as you can see here, under glasshouse condition, the lines are still in a vegetative stage, but by manipulating the photo period, the lines are already starting to set seeds, so that you can at least take up not less than 4 or 5 seasons in a year, as compared to 2 seasons, which you normally take up under field conditions. And another important tool is genome editing. And what is genome editing? In conventional breeding, if I had to improve, say, disease resistance of the recipient variety, when I transfer the gene from the donor parent, it's likely that non-favorable genes, which is called as linkage drag, are also transferred. Through this strategy also, we can develop better varieties. When used molecular markers, conventional breeding becomes more precise. And genetic engineering is where, from another variety or from another organism, we clone the gene and then transfer it into a new variety. In this, only the new gene is introduced. So this is slightly better than conventional breeding. But the best tool is called as genome editing, in which precisely identify a susceptible variety and identify the locus which is associated with disease resistance, or in terms of disease susceptibility. So edit that susceptibility gene through tool called as CRISPR-Cas9 mediated gene editing. Now that variety will have small changes in that particular susceptibility locus. So after this changes, the whole new variety will be the same like the old variety, except for the fact that it will be disease resistant. And this tool is very, very powerful because it is rapid and then targeted approach, unlike conventional breeding or marker-assisted breeding. Product delivery can be done within 3 to 5 years, which generally is not the case in genetic engineering because of regulatory issues. It takes anywhere between 8 to 15 years to deliver a product. And some breeders are saying it can be a future of plant breeding also. So through this tool, my colleague Dr. Satyendra, by taking cues from his popular paper, Ashikari, wherein he has identified a major gene called as GN1A, and he has identified that cytokinin oxidase is the gene which is responsible for GN1A. If a mutation is there in cytokinin oxidase gene, its function is abrogated, because of which cytokinin degradation is reduced. So at this stage, Ashikari identified that in case of GN1A, the candidate gene is called as cytokinin oxidase. Because of natural mutations in the variety Habataki, cytokinin oxidase gene function is impaired. And because of this, Habataki has got increased number of grains. Because more cytokinin accumulates in the grain, which leads to indirectly to more number of grains. And based on this, we thought that by modulating cytokinin levels, we can possibly increase the grain yield and possibly ushering new green revolution. And we have been successful to a significant extent also. This principally based on a discussion between Dr. Ashikari, who published this science paper, and my colleague, Dr. Satyendra Kumar Mangrotia. If Dr. Ankur, if you are interested, I can request my colleague, Dr. Satyendra, at a later stage to give a lecture specifically on gene editing and its prospects and what.
Definitely, Doctor. Definitely.
I'll pass on his contact details. You can contact him.
Yes, please, please. Yes, sir.
डॉक्टर सत्येंद्र का खूब ये खासियत है कि जिनको हिंदी में चाहिए वो मैसेज हिंदी में भी डिलीवर कर सकते हैं. He's a champion in delivering the right message in local language also. So Dr. Satyendra's efforts, what he did is he edited not the original locus, originally in this variety Habataki, the gene, the mutation had occurred naturally on chromosome on exon 1. What Dr. Satyendra identified is that mutating exon 3 or exon 4 will be more effective. If anybody is interested, I can explain it. Very shortly, a paper is coming out. And then through that, he developed a mutant version of the variety Samba Masuri, which I also manipulated for bacterial blight resistance. Through this particular mutation, he's now able to develop a better version of cytokinin oxidase gene, wherein editing is the abrogation of function is much stronger. So what has happened is that as compared to BPT 5204, which is Samba Masuri, the genome edited version, which is also called as DRR than Kamla, 100 or Kamla, wherein there is no transgene is there, only a gene edit is there. This is now able to yield at least 25 to 40% more yield than Samba Masuri. And what we observed is that this line has got better photosynthetic efficiency and better root system. Because of that, it also matures early, at least by about 20-25 days, because of which it is able to save at least 1 or 2 irrigations. And it still retains the same grain and cooking quality like the original variety. And most importantly, it is not a transgenic variety. Even though the transgenic process was used in generating Kamla mutant, through the process of meiotic recombination, the transgene was removed. And as of now, Kamla doesn't have any transgene. It only has a mutation in the gene called as cytokinin oxidase, for which a lot of natural mutants are also available. And as you can see here, because of better photosynthetic efficiency, it has got a better culm or stronger culm, so lodging will not be there. Increased panicle size, and surprisingly, we also observed that it performs very well under drought and then reduced nitrogen applications. And this line has undergone 2 years of trials across 30 locations in the country. And based on its superiority, it has been identified for varietal release. And the variety has been released by our Honorable Minister of Agriculture. Now, let me move to another important aspect, the last topic in which I'll be talking briefly, called Artificial Intelligence, how we can make a difference in plant breeding. For those who are interested to know more, this is a review paper written by Farooq et al. In Trends in Genetics. It's available free of cost. The title of the paper is A Roadmap for Artificial Intelligence Enabled Plant Breeding. So what do we do in artificial intelligence? We generate a lot of data in terms of gen bank accessions or gene bank accessions, and also through speed breeding, mainly through breeding materials, wherein we have got a lot of genotype and also phenotype data generated over the years by different groups. So what we essentially do is through techniques like multi-omic tools, mass spectroscopy, sequencing, testing in multiple environments, by phenomics, drone, sensor-based thing, we generate big biological data from breeding materials and also gene bank materials. And what we try to do is, we try to identify market trait linkages, wherein we identify the genetic networks, and also we try to predict the phenotype. This process can be accelerated by deployment of tools like a smart breeding platform, which can automatically analyze and then identify the set of parents which can be used for crossing, and a set of gene targets for genome editing, and also help in identifying of better lines even without phenotyping. This has been deployed in a few crops like maize and tomato, and we are trying to deploy it in crops like rice, wheat, and all. There's a lot of potential, not limited to what I explained. There are several reviews, but I think in terms of making sense out of big biological data, and also in terms of phenotype prediction, and in terms of identifying the right set of lines or targets for gene editing, I think artificial intelligence will be very, very useful. And particularly generative artificial intelligence and machine learning will be the future of plant breeding. And this is not only limited to genetics, even designing the breeding program, designing the process also, artificial intelligence will be very, very useful. And applications of AI include genomic prediction or selection, trait prioritization, which is a step done before crossing, QTL detection, wherein QTLs can be detected very, very fast by algorithms, and optimizing marker-assisted selection and genomics-assisted selection. And what we have seen is genomics-assisted breeding, we have seen the phase 1. But what we are talking right now, through integration of haplotype-based breeding, gene editing, and also through artificial intelligence and then big data, we are marching towards genomics-assisted breeding too, which is aptly described in this paper written by Tyagi et al. In Agronomy. This is also freely available. And what I believe is that through the development of improved Samba Masuri, we have seen this is a collaborative work wherein not a single institute is involved, multiple institutes, at least 2 institutes are involved. We are collaborating for the last 28 years. When farmers approached us, together with Dr. Amish Sonti's group, we identified the right set of genes and we pooled all our resources. And then through a collaborative effort only, this was possible. And improved Samba Masuri is not only helping farmers to reduce the disease incidence, it's also giving added benefits to farmers. A socioeconomic survey done in 2017 by Manage has shown that improved Samba Masuri has clear advantage to farmers, helping them to earn in the last 10 years more than 1,200 crores. In this, what I mean to say is right selection of the target genes are important. And also I would like to tell that marker-assisted selection or genomics-assisted selection can deliver products quickly at a lower cost. And the third thing what I would like to say is, once you develop a good product, you don't stop with that. Try to add value to that product. So multi-trait packaging through genomics-assisted breeding is the only way for developing climate-resilient rice. And what we have seen is that genomics is a really good tool. And what we need is detailed characterization of a greater number of economically important genes, which are relevant to India is required. Presently, the progress regarding structural genomics in terms of sequencing is going on very fast, but identifying the function for each and every gene is not very good. We need to accelerate our efforts. And new tools like gene editing, speed breeding, and haplotype-based breeding, and application of artificial intelligence can transform breeding, and it can improve several recalcitrant traits which are part of climate-resilient traits. And I would like to present a proof that climate resilience, climate rice production in India is really climate resilient, because we had the years like 2001, 2002, which are typical year, like a normal abnormal year. 2008, 2009 is also normal abnormal year. But here you can see, even in by the every time abnormal year happens, there will be a decrease in rice production in terms of million tons. Starting with 2020-21, we have clearly seen that even after 3 abnormal years wherein monsoon has failed or monsoon has been spread has not been very uniform, our rice production has steadily increased. This possibly because of the climate resilient rice varieties and mainly because of efforts of our farmers who have wholeheartedly accepted these climate resilient rice varieties. And I think this is a step in the right direction. I would strongly say that based on this particular proof that genomic assisted breeding has accelerated development of climate resilient rice varieties and hybrids. And we should also think about a question. As a breeder, he has multiple tools available with him. And biotechnology is only one among the many tools available to a breeder. If a breeder asks a question whether can he use tools like biotechnological tools like genomic assisted breeding for accelerating the breeding, that's true. But if the breeder tells that is biotechnology only the tool available, I would say no. Biotechnology or genomic assisted breeding is one of the tools which can complement and supplement the process of breeding. It can never replace breeding. So I would say a new breeding strategy called as convergence breeding, which combines both conventional breeding along with tools like genomic assisted breeding, is the need of the hour. And before I finish the presentation, I would like to thank all my colleagues who have contributed. And I would also like to thank all the funding agencies which have been behind us in terms of supporting us in these efforts. Thank you very much. Now the forum is open for any questions or comments if you have.
अब ये 10 मिनट का डाउट क्लियरिंग सेशन है। आप सभी पार्टिसिपेंट्स को पता है इस पर्टिकुलर टॉपिक को लेके जो प्रेजेंटेशन अभी दिया गया है डॉक्टर साहब के द्वारा, इसके अलावा भी यदि राइस से रिलेटेड आप कुछ जानना चाहते हैं, आपका बहुत अच्छा एक्सपीरियंस है और भारत के बहुत पॉपुलर राइस ब्रीडर हैं आप, काफी प्रजातियां आपने डेवलप करी हैं, तो जो भी पार्टिसिपेंट्स कोई भी क्वेश्चन पूछना चाहते हैं उनका स्वागत है। और सभी फैकल्टी और साइंटिस्ट हैं। 200 से ज्यादा पार्टिसिपेंट्स को मैंने देखा था, बरसात के कारण शायद 150 प्लस रह गए हैं। तो आपसे रिक्वेस्ट है कि प्लीज आप कोई भी क्वेश्चन जो रिलेवेंट क्वेश्चन है वो आप पूछेंगे, भारतीय राजनीति से जुड़ा हुआ नहीं, जो भी आपका राइस से रिलेटेड है आप पूछिए।
आप कोई भी सवाल पूछ सकते हो, जिसका जवाब मैं जानता हूँ आपको अभी दे सकता हूँ, जिसका जवाब नहीं जानता आपको रीडायरेक्ट कर सकता हूँ किन से उसका जवाब मिल सकता है,
जवाब मिलेगा। जी जी जी।
और जी मैं आपको
हेलो सर,
इसका पीडीएफ कॉपी देता हूँ so that you can distribute it freely to
हेलो
डेफिनेटली सर।
remind me because I may forget it. Just if I don't send it by tomorrow, आप थोड़ा मुझे रिमाइंड कर लीजिए और मैं
जी सर।
आप और सवाल आप हिंदी में भी पूछ सकते हैं, इवन तो मैं स्वाभाविक तौर से आप मूल रूप से हिंदी भाषी नहीं हूँ, बट थोड़ा बहुत जानता हूँ, मैं आपको जवाब हिंदी में भी दे सकता हूँ।
लास्ट मीटिंग भी सर जो नास में हुआ था, मंत्री जी के साथ आप थे, तो आपने पहले ही बोला था कि मैं हिंदी में इतना कंफर्टेबल नहीं हूँ, लेकिन मैं अपनी बात रखूंगा। ठीक है, प्लीज कोई साइंटिस्ट बात करना चाहे तो प्लीज बोलिए।
हेलो सर,
बताइए।
सर एक्चुअली मैं जलंधर पंजाब से बोल रहा हूँ सर, लवली प्रोफेशनल यूनिवर्सिटी। तो सर यहाँ पे अभी क्या है, फ्लड का सिचुएशन है लाइक काफी, और बच्चों के सर ट्रायल्स लग रखे हैं लाइक राइस पे। तो सर क्या हुआ कि पिछले एक दिन से ना वो ट्रायल्स जो बच्चों के हैं वो सबमर्ज्ड कंडीशन में है, लेकिन विद इन लाइक 2 और 3 डेज पानी सर उतर जाएगा उसमें से, फील्ड में से निकल जाएगा। तो सर क्या वो राइस सर सर्वाइव कर पाएंगे या वो ग्रोथ आ पाएगा, या फिर लाइक मतलब कैसे उस चीज से डील करें सर?
एक बात पूछना चाहता हूँ कि
जी सर।
ये जो सीडलिंग जो लगाए हैं आप
यस
ऋषभ जी, आप ऋषभ जी अच्छा रोहित ठाकुर बात करें।
हाँ जी सर।
जी आप बताइए कि कितने दिन का ट्रांसप्लांट?
21 दिन पे ट्रांसप्लांट कर दिया था उसको, लाइक वेराइटी PR126 थी सर, लेट वेराइटी लगाई है। और अभी रिसेंट उसका डाटा दिया था बच्चे ने सर, उसका डाटा था, उसका हाइट लगभग मान के चले सर 60 सेंटीमीटर अबाउट था उसका हाइट अभी।
ठीक है, अभी ज्यादा वरी करने का जरूरत नहीं है, क्योंकि जब बहुत छोटे सीडलिंग स्टेज में है, ट्रांसप्लांटेशन करने के 10-15 दिन के बाद थोड़ा स्ट्रेस रहती है, थोड़ा बड़ा हो जाए, पानी में जाए 2-3 दिन के अंदर, तो कोई डरने का जरूरत नहीं है। फ्लावरिंग डिले होगा आपको, लेकिन उसमें यील्ड में कोई कमी नहीं आएगी और अच्छा फसल आएगा आपको।
ओके ओके ओके, क्योंकि सर सब मर्ज्ड हो रखा है यहाँ पे सारा ना, तो बच्चे थोड़ा डर गए हैं कि ट्रायल फेल हो जाएगा, रिपीट होगा, तो एक पैनिक सा
नहीं
वो
नहीं
चीज।
नहीं, जितना
ओके
हो
ओके
सके आप थोड़ा जल्दी ड्रेन करने का कोशिश कीजिए।
जी
बाकी
सर।
2-3 दिन का सबमर्जेंस मामूली सी बात है, कोई इफेक्ट नहीं होगा। यदि उसमें दाना नहीं निकले तो कोई प्रॉब्लम ही नहीं है। दाना निकले तो थोड़ा डैमेज होने का चांसेस ज्यादा है। दाना नहीं निकले तो बिल्कुल ठीक है, कोई प्रॉब्लम नहीं।
ओके ओके ओके, थैंक यू सर।
नेक्स्ट प्लीज।
हेलो
नेक्स्ट।
हेलो डॉक्टर साहब।
बताइए सर।
हेलो
सर
सर मेरे को
सुना
ये
ना सर?
बताइए। हेलो
बताइए सर।
डॉक्टर साहब, जब और प्लांट गल जाते हैं पानी में
हाँ सर।
तो राइस को ये क्यों नहीं गलता? क्या ऐसा होता है इसमें? लेयर और सिलिकॉन की सुना है मैंने होती है। हाँ, प्लीज क्लैरिफाई इट।
सर उसमें ये दो बात होती है सर। फ्लड्स में
जी
सिर्फ पानी आती है, चावल
जी।
सबमर्ज हो जाता है, थोड़ा अच्छा हाइट है, सबमर्ज हो जाती है, 2-3 दिन में पानी निकल जाता है तो कोई प्रॉब्लम नहीं है। लेकिन ऑफन
ओके
देखते हैं कि जब बाढ़ आ जाती है तो पानी के साथ साथ रबिश भी आ जाती है और कीचड़ भी आ
ओके।
जाती है। जब कीचड़ डिपॉजिट हो जाती है तो उसको वो एक लेयर टाइप में फॉर्म हो जाती है और
ठीक सर।
थोड़ा रोग लगना या सड़ जाने का भी संभावना है सर।
ओके, इट कैन रिकवर्ड।
या यदि सिर्फ पानी का ही है और बहता हुआ पानी
जी
है तो प्रॉब्लम नहीं है। थोड़ा कीचड़
ठीक
के
सर।
साथ सबमर्ज पानी है तो उसकी वजह
ओके
से थोड़ा यील्ड अफेक्ट होगा सर। लेकिन मामूली सा 2-3 दिन का तो कोई प्रॉब्लम नहीं है। इसीलिए बोलते हैं
ओके
राइस
सर।
भी कल्टीवेट करते हैं, ज्यादा
जी
डीप नहीं लगाना। अच्छा ड्रेनेज जहाँ पे भी हो वहीं पे लगाने के लिए हम लोग रेकमेंड करते हैं सर।
ठीक सर। ओके, थैंक यू
आपका
सर।
कौन सी
थैंक यू।
एरिया का बात कर रहे हैं सर? कहाँ पे सबमर्जेंस हुआ सर?
जल जलंधर पंजाब।
जलंधर पंजाब। तो जलंधर
जी
पंजाब में तो दोआब एरिया में आता है, तो वहां पे तो ड्रेनेज अच्छा हो जाना चाहिए। काफी
फिर
दिन
अभी फ्लडिंग कंडीशन है ना, जैसे पहले बता रहे थे रोहित जी। फ्लडिंग कंडीशन है।
फ्लडिंग कंडीशन है सर, बट
जी सर।
अभी फ्लड तो रुक गया ना, मतलब पानी का बहाव
सर बारिश हो रही है अभी भी ना?
अभी भी हो रही है, तो थोड़ा
जी
देखना पड़ेगा सर। 4-5 दिन से ज्यादा समझ रहेगा, राइस पे क्षमता नहीं है। इवन एडल्ट
जी
प्लांट
सर।
स्टेज में भी 4-5 दिन से ज्यादा रहेगा तो सड़ना शुरू हो जाएगा सर।
ठीक सर। ओके, थैंक यू।
थैंक यू सर।
नेक्स्ट प्लीज।
हेलो सर।
हेलो
बताइए सर।
हेलो सर जी, मेरा नाम डॉक्टर ऋषभ मकवाना है। मैं वडोदरा गुजरात आनंद एग्रीकल्चर यूनिवर्सिटी से हूँ।
बताइए
ये
सर।
मेरा क्वेश्चन रिगार्डिंग सॉइल से था।
बताइए।
एक्चुअली क्या है कि सर यहाँ पे ज्यादातर फार्मर्स का क्या रहता है कि राइस करना है तो वो थोड़ा पानी भरा हुआ रहना चाहिए, जैसे आपने अभी कहा कि ड्रेनेज अच्छा होना चाहिए। तो क्या हम रेकमेंड कर सकते हैं ये रेड सॉइल या गोराट सॉइल, जहाँ पे क्या होता है कि वो उसमें ड्रेनेज काफी अच्छा रहता है। और कई जगह पे लोग क्या है कि अच्छी जमीन भी हो तो उसमें भी राइस प्रिफर करते हैं जहाँ पे हॉर्टिकल्चर प्रोड्यूस कर सकते हैं। सो इज इट बेटर टू एडवाइज हॉर्टिकल्चर क्रॉप्स और इट इज बेटर टू एडवाइज राइस क्रॉप? और कौन सी
सर
टाइप की सॉइल में ज्यादा बेटर रहेगा सर?
सर एक बात है, राइस का अच्छा क्षमता है, स्टार्टिंग फ्रॉम एसिडिक सॉइल्स टू अल्कलाइन सॉइल्स, इन सलाइन और सोडिक सॉइल्स आल्सो राइस ग्रो कर सकते हैं। तो सॉइल का कितना है, थोड़ा सॉइल का जांच करवा लीजिए, इसका पीएच और ईसी, इन दोनों का जांच के बाद आप उस पर डिसाइड कर सकते हैं कि सॉइल यदि सलाइन और अल्कलाइन प्लोन, तो हॉर्टिकल्चरल क्रॉप ज्यादा बढ़िया ग्रो नहीं करेगा, एक्सेप्ट अमरूद या चीकू, ये सभी ग्रो कर सकते हैं, बट बाकी क्रॉप्स ग्रो नहीं करेगा। राइस के लिए जहां तक सवाल है, वेल ड्रेन सॉइल चाहिए और साथ ही में ये भी सवाल दिखना चाहिए कि वेदर वी रिक्वायर मोर एरिया अंडर राइस, क्योंकि अभी जो प्रोडक्शन है, हम लोग जो हमारे देश का जो जरूरत है, मान लीजिए करीब 90 मिलियन टन तक जरूरी है। बाकी जो भी प्रोड्यूस कर रहे हैं वो बफर स्टॉक में जाती है और उसमें कुछ 25 मिलियन टन एक्सपोर्ट करते हैं। सो आपको ये देखना पड़ेगा कि उस पर्टिकुलर एरिया में मार्केट है चावल के लिए, तो वहां पे रेकमेंड कर सकते हो सॉइल के अलावा। क्योंकि जब भी हम रेकमेंड करते हैं, फार्मर के जो इकोनॉमिक्स के ऊपर भी ध्यान देना चाहिए और डिमांड के ऊपर भी है सर।
सर वन एडेड क्वेश्चन टू दिस, अगर ये पीरियोडिकली काफी साल तक राइस ग्रो करते हैं तो क्या उसके सॉइल पे एसआरडीएस इफेक्ट या कोई न्यूट्रिएंट डाउन पे होता है ऐसा कुछ असर?
हाँ सर,
ये
जब
थोड़ा एक मिथ है हमारे यहाँ। थैंक
इसीलिए
यू
हम
सर।
बोलते हैं कि राइस के बाद कोई रोटेशन रहना चाहिए। रोटेशन मतलब कोई पल्स का हो या ऑयल सीड का हो, जो सॉइल का न्यूट्रिएंट स्टेटस बढ़ा ले, तो बहुत बढ़िया है सर। कंटिन्यूअसली राइस आफ्टर राइस दोनों सीजन में ग्रो करें तो खेती के लिए अच्छा नहीं है सर, रेकमेंड नहीं करते हम लोग।
थैंक
नेक्स्ट
यू सो
प्लीज।
मच सर,
साउथ
थैंक
इंडिया
यू थैंक यू।
में अशुड मार्केट है एंड गवर्नमेंट भी एक्स्ट्रा प्रोक्यूरमेंट प्राइस देते हैं, उसकी वजह से राइस राइस ग्रो करते हैं। जैसे पंजाब हरियाणा में राइस भी ग्रो करते हैं, जो सॉइल के हिसाब से अच्छा नहीं है। बट क्या करें, फार्मर भी तो जब व्यापारी को अपने प्रोड्यूस अच्छे मार्केट में सेल करने का ये है, फार्मर भी सोचेगा कि एक सेफ क्रॉप है राइस, उसको ग्रो करे तो अशुड मार्केटिंग भी है, तो कैसे डिस्करेज कर सकते हैं।
बिल्कुल सर, और थैंक यू सर, प्रेजेंटेशन वो बहुत ही बढ़िया था। काफी चीजें थोड़ी हमें नहीं जानने मिली। थैंक यू सो मच सर।
आपको किसी भी चीज में आपको सवाल है, मेरा नंबर तो अंकुर शर्मा जी के पास है, उनसे नंबर
जी
ले
सर।
लीजिए, नहीं तो मेरा
जी।
ईमेल आईडी नोट कर लीजिए, rmsundaram34@gmail.com, rmsundaram34@gmail.com। इस पे सवाल भेज दीजिए, जिनका जवाब मैं जानता आपको इमीडिएटली दे दूंगा, जिसका नहीं जानता पता करके बता दूंगा या रीडायरेक्ट कर दूंगा, ठीक है।
जी। कोई
और
और साइंटिस्ट और कोई फैकल्टी यदि कुछ बात करना चाहते हैं तो प्लीज।
मैं कुछ चैट बॉक्स में भी कुछ लोगों का कमेंट देख रहा हूँ। हालांकि कोई स्पेसिफिक कमेंट या ये नहीं है इन टर्म्स ऑफ आस्किंग। अच्छा, डॉक्टर संगीता सावंत ये सवाल पूछ रहे हैं कि इज इट पॉसिबल टू एडिट मोर देन 2 जीन्स विद जीनोम एडिटिंग? हाँ संगीता जी, जब डॉक्टर सत्येंद्र प्रेजेंट करेंगे, वो अपने एफर्ट प्रेजेंट करेंगे, अप टू अबाउट 7 or 8 different genes also it is possible to edit. For example, in a local aromatic rice variety called Chitti Mutyalu, Dr. Satyendra has edited at least 5 different genes. It is indeed possible. And can we cultivate Samba Masuri in hill areas? नहीं, क्योंकि उसका जो दाने का जो टाइप है बारीक है, और इसका छोटा आकार का है। हिली एरियाज में जनरली देखा हूं कि जो दाने का जो है मोटा दाना या जो थोड़ा लंबा दाना प्रेफर करते हैं। आपको यदि हिली एरियाज के लिए कुछ वैरायटीज चाहिए तो मैं रेकमेंड कर दूंगा। वैसे 2 संस्थान है, एक हिमाचल में रीजनल एग्रीकल्चर रिसर्च स्टेशन मलान करके, सीएसके हरियाणा, सॉरी हिमाचल प्रदेश एग्रीकल्चर यूनिवर्सिटी में है। दूसरा एक
पालमपुर।
संस्थान है वीपीके अल्मोड़ा, विवेकानंद पर्वशीय कृषि अनुसंधान संस्थान करके, वहां से पूछ के आपको मैं वैरायटीज बता दूंगा, ठीक है, सुटेबल वैरायटीज। और और कोई सवाल तो नहीं है?
नहीं, कोई। डॉक्टर रितेश कुमार जी, आप पूछना चाह रहे थे कुछ?
आप बताइए सर, पूछिए सर। रितेश जी पूछिए।
डॉक्टर साहब, नेटवर्क का कुछ ग्लिच है रितेश सर आपकी साइड से, आप प्लीज चैट बॉक्स में लिख दीजिए, वो ज्यादा कन्वीनियंट रहेगा। कोई बात नहीं सर, आप परेशान मत होइए, ईमेल आपको मिल गया है, आप ईमेल कर सकते हैं या मुझे क्वेश्चन दे देना, मैं सर से रिप्लाई ले लूंगा। कोई और फैकल्टी और साइंटिस्ट पूछना चाहे प्लीज। ओके, ठीक है, नंदिनी जी।
So everyone is सारे तारीफ ही कर रहे हैं सर की, और कोई क्वेश्चन तो नहीं है किसी का। So,
जी।
on the behalf of organizing committee of this FDP training program and AgriMeet Foundation Bharat, I take this opportunity to express our deepest gratitude to you, sir, for delivering such an enlightening and inspiring session today. Your lecture was not only rich in knowledge but also highly motivating. The way you shared your journey in rice biotechnology, molecular breeding, and the development of improved varieties has given us valuable insights into the true meaning of research for society. Your emphasis on bridging lab to land and your practical examples of how innovations like improved Samba Mahasuri and other MAS derived rice varieties are benefiting farmers will remain an inspiration to every participant here. We are especially thankful for the clarity with which you explained complex scientific concepts, making them accessible and engaging for all of us. Your session has given our scholars, faculty members, and young researchers a fresh perspective on how science and innovation can create a long lasting impact when driven with vision and dedication. It has been truly an honor to have you with us, sir. Your words of wisdom will continue to guide us in our academic and professional journeys. We sincerely hope that this is just the beginning of a continued association, and we look forward to many more opportunities to learn from your vast experiences in the future. On the behalf of everyone present here, I once again extend our heartfelt thanks to you, sir, for gracing this program and making it so meaningful. Thank you so much.
आपका भी और अंकुर जी का भी ये मौका
बहुत
देने
बहुत धन्यवाद सभी पार्टिसिपेंट्स को। नवंबर के विंटर स्कूल में भी सर का लेक्चर फिर से मिलेगा।
जरूर जरूर
बहुत
सर।
बहुत धन्यवाद कि सर आपने मेरी रिक्वेस्ट को एक्नॉलेज किया और अपना टाइम निकाला। थैंक यू सो मच। जय श्री राम। जय श्री राम।
मुझे भी बहुत आनंद हुआ सबसे इंटरेक्ट करने में। थैंक
जी
यू। थैंक यू
जी। गुड नाइट। गुड नाइट।
थैंक यू सर।
जी सर। जी सर।
धन्यवाद। थैंक यू।
जी जी।